Chinese Academy of Science with research interest in identification and selection of genes associated with agronomic traits in domesticated plants and animals, and using investigation of domestication history by population genetics and evolutionary genomics approach. Please welcome Lu Jun.
Good afternoon, ladies and gentlemen. It's a great honor to introduce about my work here. First, I would like to thank the organizer for candidly offering me a chance to speak. Today, my topic is identification of elite variety tag SNPs, and I would like to call them ETAs size in my study. It is a new approach for identifying the loci underlying crop improvement. Okay, as we know, we have many accessions in rice. We have the white accessions, the land races, and we also have some elite varieties with very good performance. And many elite varieties probably contain some elite alleles that contribute to their superior performance. And by elite alleles, I mean the alleles that probably contribute to the superior traits of crops, such as good yield or good eating quality, and resistance to biotic or abiotic stress. And usually we can use these two approaches to identify those elite alleles. They are both based on linkage and recombination. Using QTL mapping, we have mapped a lot of genes. This method has been proved to be efficient. However, it is labor intensive and usually requires lots of time to construct the segregating populations. And on the other hand, the association mapping, another popular method, have very good resolution, providing you have a large population, and also can save a lot of time. But it tends to miss the elite alleles because usually the elite alleles can be very rare. Okay, so how to identify the elite alleles that are probably present at a low frequency in the gene pool? 3 years ago, when I began my research as a graduate student, I began to think about that question. And I think maybe the idea can be straightforward. By comparing the genome of elite strain with the non-elite populations, probably I can identify some clues, identify those elite variety tag alleles or elite variety tag SNPs that are fixed in a particular elite strain, but are at low frequency in the non-elite populations. So, do we have the non-elite populations? The answer is yes. Fortunately, we can have access to many non-elite the genomic data of non-elite accessions. For example, in the last year, our lab published a data set including 50 accessions of land races and white accessions. And the year before last year, Professor Binhan's group published a very huge data set consisting of 517 Chinese land races. And those are all unimproved strains belonging to the non-elite accessions. And I will use them in my data analysis, and I call them control panel 1 for this data set and control panel 2 for this data set. Okay, then I sequenced some elite varieties. I chose 6 elite varieties based on their agronomical importance. For example, I have Indica Guichao 2, which has extraordinary high yield, and also a Japonica Irad 104, a very famous upland rice for its adaptation to the upland environment and also very strong drought resistance. And also I have Koshikari, a renowned Japonica with exceptional aromatic smell. Then I use the next generation sequencing to sequence each variety in 15x coverage, relatively deep sequencing. And I chose 5 individuals for each variety in order to eliminate the within variety polymorphisms. Okay, after getting the sequencing data, I compare the genotype of a specific elite variety with a non-elite population and identify those elite variety tag SNPs for each variety. I set the threshold as 5%. Here, this picture shows the ETAs distribution for the Indica Minghui 63 and Japonica Erat 104. And on average, we identified around 10,000 ETAs size for each variety. And we can see different elite varieties have different ETAs distribution pattern. That is because they are bred for different purposes. So when you look at the genome, you will find different targets, right? Okay. Because previously many studies identify the mutation, the SNP mutations. For example, the non-synonymous mutation, premature stop codon, or splicing defect. This kind of mutations can confirm the change of agronomical trait. So I went further to use the genomic annotation information to assign those ETAs sites into different genomic regions, and to pick up those causing non-synonymous mutation or gene structural disruption. And I call them large effect ETAs. And for each elite variety, we identify several hundred of large effect ETAs. And these sites have great potential to alter the gene function, and thus have may probably be involved in the in impacting the agronomical phenotypes. I think this table provide a valuable checklist for the following up functional studies. Among those large effect ETAs, we notice a very interesting large effect ETAs. It is a non-synonymous mutation in the upland rice ERAT 104. It is a mutation from C to T, which means the upland rice has a unique T genotype. It is located in a HYD gene, and this gene encodes a critical enzyme for ABA synthesis. By in silico prediction, we found that this mutation probably changes the secondary structure around the enzyme binding site, which is a strong indication that it may probably contribute to the alteration of the enzyme activity. As I have mentioned, these ETAs sites are to some degree they are elite variety specific. So they are present at a low frequency in the control panels. Then we look into detail about the genotypes in the control panels. We found that in control panel 1, all of the 61 accessions have the C type, and in control panel 2, which consists of 517 accessions, 38 of them have T genotype, and we were very surprised to find that 37 of the 38 are actually upland rice. So this evidence strongly suggests that this T genotype may be associated with upland adaptation. Therefore, we went further to expand our sample size by choosing 109 upland accessions and 102 irrigated accessions to genotype this site, which results in an allele frequency of 61% of the T genotype in upland population, but no more than 3% in the irrigated population. So you can see this is a huge allele frequency difference. Then we went further to see whether we can observe any selection signal around this region. Using resequencing data, we calculate the nucleotide diversity level around this region. As you can see in this figure, the red line refers to the T-type upland population, The blue line refers to the C-type upland population, and the black line refers to the irrigated population. We can see there is a obviously low diversity region around this ETA size, indicating that there may be a selective sweep here. Then we also use the phylogenetic analysis. Here, in this picture, the blue branches indicates the T-type upland rice, while the green branches indicates the C-type upland accessions, and the black is irrigated rice. And when we use the 350 kb sweep region to construct the phylogenetic tree, we found that all of the T-type upland accessions cluster together to form a monophyletic tree. When we use its flanking region, we found the T-type upland accessions become scattered. So this results indicates the 350 kb probably have a distinct genealogy, and which means it may probably be selected in the upland rice breeding. Okay. There are in total 11 genes within this 350 kb region, and in this region we only identified 2 large effect ETA size. Besides the one I have mentioned in the HYD gene, we notice there is another mutation which causes us 28 amino acids truncation from the C terminal. However, this ETA size only has an allele frequency difference of 23%, much less than that of the ETA size in the HYD genes between upland and irrigated population. So we thought the other large effect ETA size may probably be hitchhiked with the one in the HYD genes. Okay. As I have mentioned, the HYD genes encode a very critical enzyme for the ABL synthesis. So we were wondering whether a functional study can provide some insight. We measured the ABL level of the T-type upland accessions and C-type upland accessions, and we did observe a significantly upregulation of the ABL level in the T-type upland accessions. And we also have a recombinant inbred lines population. And after measuring the ABL level in this population, we got a consistent result. The T-type families has significantly higher level, ABL level than the C-type family. And ABA, as we know, is a very famous stress hormone and has been proved to participate in the drought tolerance, drought resistance by many studies. And as Professor He told yesterday, ABA also plays an important role in the lateral root branching. We also investigated the phenotype of lateral roots, and we found that the T-type upland rice seems to have better developed lateral roots compared with than the C-type upland rice. And in the recombinant inbred population, we also observed a similar pattern. We also quantified the lateral roots by counting the lateral roots numbers and divide the lateral root numbers by the main roots lengths so that we can get the lateral root density. We observed that T-type upland rice has significantly more lateral roots than the C-type upland rice. And in the recombinant inbred population, we got a consistent result. So taking this together, we identified the allele in upland rice ERAT104, and actually this allele has been probably selected to a high frequency in the upland rice, and because it can contribute to the ABA increase and simultaneously promote the lateral root branching. And we guess this adaptive phenotype may also contribute to the ultimate yield, because the yield is the ultimate phenotype that farmers selected. And after searching the literatures, we found there is a large effect QTL which colocalizes with our ETAs, and this QTL is so far the only large effect repeatable QTL for grain yield under drought stress in upland rice. So I think this is also good evidence for the function of this ETA size. Okay, to sum up this speech, I think we have proposed a new concept of elite variety tag SNPs. Although this method is simple, but I believe it will prove useful in the future breeding, because sometimes the useful method is not necessary to be complex, I think. And also, we give a case study which demonstrates the efficiency of this method. Moreover, I think the elite SNP alleles that we identified in upland rice may be promising for the future upland breeding. Okay. Finally, I would like to thank Professor Wen Wang and Professor Fengyu Hu. They are my supervisor. And I thank Professor Henry Nguyen from University of Missouri, who give very constructive advice about the adaptation, the rice upland adaptation. And also thank Professor Ken McNally from Erie and Professor Xiongli Zhong from Huazhong Agricultural University for their kind help. Okay. Thank you for your attention, and any question is welcome. Thank you.
Thank you. Any question?
Okay.
Hey.
Yeah. 15x. Yeah. Basically, we didn't define haplotype. We just scanned the genome and SNP by SNP. We didn't define the haplotype. Yeah. Okay, any more questions?
Maybe one question. I try to follow you. Seems like you knew already the candidate gene that you want to look at it. And when you survey across population, then you see some association, right?
Pardon?
Would it be another way that the candidate gene approach, would that be comparable to what you're doing?
Yes. Candidate in our analysis we did use the knowledge about the upland adaptation, but I think without the ETAs analysis, it's very hard to catch that mutation. Candidate, I just used the one case to demonstrate that we may find some targets, we may find some really important alleles in those lists. And for the other alleles, we think maybe there are some alleles may be also important. We are currently conducting some transformation experiments to illustrate their functions. I think functional experiments is needed, and if you have knowledge of candidate genes, this will help you. But if you don't have the knowledge, I think it is possible to identify some new alleles with our approach. Yeah.
Any more questions? So
I'm assuming that you're just presenting one of the example from your analysis, right? And could you tell us like what are the traits that you are undergoing on the investigation at the moment from your analysis?
This is a good question. Why I focus on that case? That is because upland adaptation. We have some previous work about upland adaptation, and we are familiar with this phenotype. But for the other phenotypes, for example, if you have a lead variety which has wide adaptability, which probably means that this variety, which variety can have the heading date of that variety can, maybe you can use the heading date as a phenotype. In one word, you need to know the phenotype of the elite variety that you really care, and then you using the ETAs analysis and combine with your knowledge of the phenotypes, you narrow down the mutations that, yeah. Thank you.
Anyway, we wish to see your complete work in India next year. Okay.
Thank you.
Please give him back a big hand.